Today I'm recording from the Center for Advanced Brain Imaging, a center that's jointly directed by Georgia State University and Georgia Tech. And we're talking about the methods that are used in biopsychology, in cognitive neuroscience. And I have with me today Dr. Audrey Duarte, a professor at Georgia Tech and also a cognitive neuroscientist. And she's going to tell us about some of the techniques she uses to look at the relationship between the brain and behavior. Dr. Duarte, glad you could join us.
Yeah, thank you.
Tell us about some of the methods that you're going to talk about today.
Okay, so in cognitive neuroscience, we have many methods that we can use to assess brain function. And we use two major methods in my lab, which are probably the two that have been researched the most. So electroencephalography, or EEG, which is sometimes called, measuring brain waves, so EEG technology, and also MRI, or magnetic resonance imaging. And MRI is a interesting technique and a newer technique relative to EEG, which has been around for about 100 years in cognitive neuroscience type of research. But MRI is a little bit newer, and they're nice techniques because they both assess brain function, but they give you complementary pieces of information. So whereas EEG gives you activity in real time as neurons are firing action potentials active, so you're measuring that to the millisecond, really. MRI has pretty poor spatial or temporal resolution, rather, but gives you a very nice idea about where in the brain activity is happening. So one technique, EEG, has great temporal resolution, and MRI has great spatial resolution, and so they complement one another.
Great. So let's talk about EEG.
Okay, so EEG, electroencephalography, is a technique in which we are recording electrical activity of many neurons, brain cells, at the same time. So one thing to keep in mind is that when we're going from brain to the scalp, which is where we're measuring EEG, there's a lot of stuff in between. So there's brain, there's cerebral spinal fluid, which protects the brain. There are these layers that protect the coating of the brain called the meninges. And then there's the skull and the fat of the scalp and the hair. There's a lot to go through. And so we can't just stick electrodes on the brain and record, get a lot of activity. We have to amplify that signal. So I'm going to show how we do that in a minute. But the first thing we need to do is get a research participant to be our volunteer subject so we can record this electrical activity of the brain. Okay, so now that we have our volunteer participant, what we're doing is we've put this cap on, and what this cap has in all of it are these little casings where we're going to stick electrodes in. And don't worry, the electrodes are blunt. They don't hurt the participant or anything like that. But what my research assistant here, Lydia, is doing is she's filling up these casings with this electrolyte gel. And what this gel allows us to do is it's one step of kind of amplification of the signal that's coming from the brain. As I mentioned before, there's a lot of stuff that this electrical activity has to travel through in order to reach the scalp, the surface of the scalp. So she's filling up these little casings with this gel, and when that's complete, we're going to put electrodes into these little casings, and then we'll be able to record electrical activity. So, for example, I have two of them here. They're just blunt on one side. Put them in. They kind of snap into place. Doesn't hurt or anything, right?
No.
Okay. And then once we've done that, we're going to go into another room, plug all of this into an amplifier so we can measure these really tiny little signals. Electrical activity of the brain is actually very tiny when it reaches the surface of the scalp. It's measurable, but we need to amplify that signal. So now we have our research participant, Michael. He's all plugged into the amplifier. His electrodes are plugged into the amplifier. And what we might do with EEG in the research setting is going to be pretty different from what we would do in a clinical setting. So EEG has important clinical uses. So for example, a patient who is epileptic and we're trying to identify when they have their seizures, how often, for things like this. You can use EEG to look at the electrical activity and see seizure-related activity when it comes on, kind of characterize it for each patient, and figure out how we're going to treat this patient. That's a clinical use of EEG. But research-wise, why we would use it is to understand, as I said, in real time, what someone is doing, how their brain is responding to a particular task. And the task that Michael is doing right here is a memory-related task. So what he's being asked to do is study lots of pictures of objects that you might see in real life, like shoes or balls or animals. And I'm asking him to decide what he thinks about these objects. Do you like the color of the object? Does it seem like a color that this object would naturally occur in? And the reason I'm asking him to do that is because I want him to remember these objects, and that's going to help him remember them, just answering this question about these objects and the color. After he's done this for a little bit, and keep in mind I'm measuring his brain activity the whole time, I'm then going to ask him if he remembers the objects that he's seen. So this is just a memory experiment. I'm interested in how brain activity can give us some information about how people are remembering. And so in this particular study, this is very interesting. What you can do is you can look at the time that people are studying the brain activity that's going on, the EEG, and you can look later at the items that they've studied, how well they remember them, and then what we can do is called back sorting, looking at the activity while they're studying and say, I know that that one is one they're going to remember, that one's one they're going to forget, and so on and so on. And so I can basically predict, based on brain activity while you study, what you'll later remember. And this is a really interesting phenomenon that's been identified using EEG activity. So this is what we're doing in this particular experiment. So what we see here are each of these lines represents one of those casings in the electrode in the EEG cap. And I've just plugged in 2 electrodes right now, and the ones that we're seeing that are plugged in are this one here up at the top and then this one here at the bottom. And the little deflections in this line that you can see, that's the EEG activity. It's positive and negative peaks and troughs in the ongoing brain activity. And all of this is representing the activity of underlying neurons in the brain. And what you're seeing when Michael blinks, go ahead and blink a few times, Michael, this is really interesting, and you can see it's really large. We don't typically want to see that with EEG though, because that's an artifact. It's something that we're going to remove from the data because it's kind of covering up the brain activity. And remember when I was talking about how tiny this electrical activity is, and it takes a lot for us to be able to measure it at the scalp, a lot of amplification. The movements of the eyes are just so much bigger. That's what pops out on the EEG. So we'll often ask our participants to relax their eyes, to try not to blink a lot, because that can be a little bit of a problem for us with EEG.
Well, we've moved from the EEG lab to the MRI lab, and MRI is a powerful tool that is used to study brain behavior, and Dr. Duarte is now going to tell us about and demonstrate for us MRI.
Great. So MRI, magnetic resonance imaging, everybody's heard about this, and you hear a lot about it in the news in particular, because one of the things that it can give us is nice, beautiful pictures of the brain while it's functioning. So we can measure the structural of the brain and just look at the picture of it. It's like a snapshot of just the structure of the brain or any other body part. But you can also look at what's called functional MRI. And here, what we're doing is somewhat similar to what we're doing with EEG, where we're looking at brain activity as people are engaged in some sort of task that's of interest to us. So for me, because I'm interested in memory, it might be a memory experiment. So I'm asking people to remember some pictures, and later I'll ask them, how well do you remember these pictures? But it could be something like emotion or language or any sort of behavioral type of task. In MRI, what we're measuring is what we're manipulating, as you might think, based on the name magnetic resonance imaging, is the magnetic properties of the body. So without getting into too much detail, our bodies are full of water. And when you put our human bodies into the MRI machine, the MRI has an extremely powerful magnet in it. It's about 20 to 80,000 times the strength of the Earth's magnetic field, which is extremely strong. So you put a body in there with subtle magnetic properties, and we've manipulated the magnetic properties of the body, including the brain, and that's going to allow us, through some complicated physics that I'm not going to go into, allows us to get a nice picture of the brain. And that's what we're going to do now with a research participant. MRI is a really safe technique. It's what we call a non-invasive technique, as is EEG. And what that means is that you're able to get a picture of the inside of the body without cutting into the body or hurting the body in some way. So whereas an X-ray can actually produce some damage in the form of radiation, MRI does not do that. So it's preferred to a lot of other methods that allow us to image the body. So one of the things that we need to do, absolutely, is make sure that our participant has no metal in her body. If she had metal in her body or on her person, that metal could be attracted to the magnet and could be very harmful to her or people that are in the direction of that flying metal object. So remember, the magnetic field of this magnet is extremely strong. So we have our research participant, Lisa, here, and she's graciously volunteered to participate in this experiment. Before we put her into the MRI machine, we need to make sure she has no metal on her. So we've asked her, do you have a metal pacemaker? Do you have any implants, like rods that you might have in a broken bone, which sometimes people get after they've broken a bone? She's said that she doesn't have any of these sort of metal implants, but we're going to check her over and make sure she doesn't have anything on her body. And this is Nitavia, our MR technologist, who's actually going to be operating the MRI scanner.
So it's going off in your lower area, so if you can remove that in your pocket. And around the metal on your boots. So we'll have you remove your boots as well before we take you in. And your watch.
So Nitavia is using a magnetic wand to make sure there's no magnetic object on Lisa's body. And sometimes shoes, as you know from traveling through airports, have metal inside of them.
I'm glad I pointed that out.
And the buttons on your dress.
So what Dr. Smith actually just very astutely pointed out is that there are buttons on Lisa's dress which she had no idea had metallic properties, and they do. So if she were to go into the MRI scanner with these metallic buttons, what might happen is the buttons could get a little bit warm, and that could maybe hurt Lisa while she's in the scanner. We definitely don't want that. We don't want to take any metal in there at all. So what we're going to have her do is go in and change and put on some scrubs like you would see on a patient in the hospital that have no metal in them at all, and then she'll be safe to go into the scanner. Okay, so now Lisa's changed into her MR safe clothing, no buttons, no metal objects on her at all. She's got her scrubs on, and what we've done is we've put her on the scanner bed. This is what the participant is going to lie on. And Nitavia is making sure she's comfortable. She's put a blanket on her because laying down for a little while you can get a little bit chilly. And the coil that Lisa has over her head, what that coil is going to allow us to do is take the pictures. Without it, we couldn't take the pictures of the brain activity. And so she has this coil on, and she's being slid now very slowly into the MRI machine. So if you hear that sound, what that sound is electricity being sent through the coil that's over Lisa's head. And what that's going to allow us to do is take pictures in 3 dimensions of Lisa's brain. And it sounds like that. She can hear it in there too, and it can be a little bit loud, so we've put earplugs into her ear so she's not hearing it quite as loud as she would be otherwise. When this is done, what we're going to get is a really beautiful picture, a 3D picture of Lisa's brain. We've now gotten this beautiful picture of Lisa's brain, and Nitavia is going to show it to us in a minute. Okay, so this is on the top left corner here. That's a side view of Lisa's brain. And this view right here is sort of a face view from the front of the face to the back of the head. And then this one on the bottom, you can actually see the eyeballs. So this is what we call it's kind of an inferior view of the brain. So these are the 3 dimensions along which we can see Lisa's brain. Now this is just what's called a structural image. It's just a picture. But what we're going to have Lisa do in a minute is tap her fingers. So we're interested in motor function and how she can perceive commands, motor commands. And so she's going to be asked to tap her fingers, and what you're going to get to see in real time is activity in the motor cortex of the brain. Now, as you can see here, we've got a beautiful 3D picture. So we're going to be able to get good spatial resolution of brain activity, but the temporal resolution of MRI, because we're actually measuring blood flow, which is a correlate of brain activity, that's quite slow, so it takes many seconds, whereas neural activity is in the millisecond time. So not great temporal resolution, but excellent spatial resolution. So should we
All right, Lisa, in just a moment, you're going to see the screen instructions change to the actual areas where you're going to tap your fingers, okay? You ready? All right, just one moment. When we're getting lines inside the block, that means she's at rest and outside of the box, and she turned to, it was programmed to match and be in sync. So she's activity, you see the peaks coming in?
Yes.
So when she's responding, the peaks should match her activity to rest. But sometimes it takes the first few seconds, it's off, and then it should co-register and be simultaneously for the red and green. Those are representing the field of view, the field of interest that I marked on green.
Okay. So over here within this green circle, there's this little green blob. And what that signifies is fMRI, functional magnetic resonance imaging activity in Lisa's left motor cortex. And the reason it's active, her left motor cortex, is because she's using her right hand. And you might have learned in class that the left motor cortex controls your right body and the right motor cortex controls your left body. So we're seeing that at play here right now. Yeah, so what we're actually asking Lisa to do is just when she sees this arrow pointing to the right, we ask her to point her right hand, and when she sees an arrow pointing to the left, we ask her to, or not point her right and left hand, but to tap her right or left fingers. When an arrow pops up, right arrow, she's supposed to be tapping her right fingers, and then that's when we're going to see activity in the left motor cortex.
Today we've learned about two techniques that cognitive neuroscientists use to study the brain and behavior. We talked about EEG and we talked about MRI. Audrey Duarte, thank you so much for telling us about these techniques.
No problem. Thank you.